Hi, my name is Steve Jackson. Thank you for joining us. I'm the Associate Director of Applications Development at Thermo Fisher Scientific in the Genetic Sciences Division. In today's talk, I'm going to tell you a little bit about how you can analyze cells that you've taken out of human subjects and manipulate them in vitro. This is important because you want to make sure the cells that you're working with are the same as the cells that you think you're dealing with, and mistakes are not made in terms of identity of those cells. So in general, this is known as human sample matching, and this is very important because the research of human development and diseases relies on the analysis of cells that you take out of human subjects and analyze them ex vivo. You put them in culture dishes or on slides, and you analyze them, outside of their normal environment. These sorts of human samples can include pluripotent stem cells, cell lines, chimeric receptor T cells, and even slices that are FFPE preserved on slides, tumor and normal matched, for example. It's critical when you study these cells and in all your studies to know that the provenance of the cells, where they've come from, and you confirm that these cells are in fact what you think they should be. It's also critical to confirm that once these cells have been manipulated outside of the subject, manipulated in vitro, that the cells have the differentiated characteristics that you want them to, and that the genomic integrity has been maintained. All these are very important for making sure that the cells are, again, correct. So there are various ways that human sample matching can be done. It involves confirming the identity of existing cell lines, and this is usually known as cell line authentication. It's important for establishing the provenance of new cell lines that you might establish in your own laboratory. You get a fingerprint of these cells so that you know that down the road those cells have maintained their identity. It's a way for tracking human tumor xenografts in mouse models. When you're doing induced pluripotent stem cell research, it's a way for matching the undifferentiated cells to differentiated cells that you have manipulated in culture conditions. It's in CAR T research, it's matching the manipulated cells to the original donor cells. And in biobanking, it's important for sample tracking and matching, as I mentioned, tumor to normal or different tissue samples from the same donor. So all of these are very important for making sure that the human samples are indeed what you think they are. And one big example of why this is important is because, as you know, there have been cell line models that have been used for human disease research for over 50 years now. And over the course of these studies, there have been 500 reported misidentified cell lines that have happened, that the cells that have been experimented on are not what the investigator thought they were. For example, there have been over 700 publications of this MB435 cell line. This was originally described as a breast cancer cell line, but it was later determined to be a melanoma. And so over 700 publications have modeled breast cancer disease when in fact it's a melanoma. So that's a big problem. And because of this, NIH and certain journals are now requiring that when you submit a grant application or when you submit a publication, that you have some kind of evidence or plan for authenticating the cells that you're using in your studies. And they want to make sure that you have a plan for making sure your cells don't become contaminated and that you're using the right cell lines throughout the course of your work. For ex vivo research, again, you must be prepared to make sure your donor and recipient of the manipulated cells match. And for biobanking, it's very important to make sure the samples are from the same individual when you think they are. So one of the ways that human sample matching is done is by making a DNA profile. This is a forensic method that differentiates two or more individuals based on their genotype at specific and highly variable loci. You probably have seen this in television programs where you do a DNA fingerprint, and it provides a very specific way to match a sample to a human donor or subject. The more loci that are used for this, the greater the power of discrimination. And so one of the most common ways to do this is shown here. There are all these different highly variable short tandem repeat sequences. I'll have more to say about them in a second, that are scattered throughout the genome. And most of the forensic identification kits make use of these different regions, between 8 and 24 of them, to come up with a molecular fingerprint that is unique for an individual. These DNA fingerprints consist of short tandem repeat sequences. These STRs are microsatellite regions of between 2 and 5 repeat lengths. They are simply repeated at random or pseudo-random frequencies in individuals, and they're highly polymorphic because they can vary quite a bit between individuals. They don't have any necessarily a functional role. They simply are length markers that are unique for an individual. The allele length is determined by the number of repeat units that are present, and so, for example, this individual has one homolog has 7 repeats on them, and the other homolog has 8 repeats. So 2 versions because they're diploid, and 7 and 8 repeats. And at this locus, then there's a fingerprint of 7 and 8 that can be analyzed. These can then, for each one of the different loci that you're analyzing, you can set up a highly multiplexed PCR reaction, and this is a standard method that it's used in forensic analysis. And the way that this works is that when you generate this PCR product, you compare it to an allelic ladder. This is an example of a photograph of the electrophoregram that comes off of the allelic ladder here, showing all the different possibilities of fragment lengths, therefore the different loci that are present. So when you then look at a sample that you want to verify, you do the PCR and run the CE experiment. Again, the electrophoregram is shown here on the right, and then you simply match up the peaks that are shown on the right with the allelic ladder that is run at the same time. And then that will give you then the different alleles that are present in the sample for all the different loci that are present in the electrophoregram. This is done automatically for you by the software, by the way. It's not something that you have to do, but the software will then be able to call out these alleles and provide for you the fingerprint, the molecular fingerprint of the sample that you're analyzing. And so when this is done with the Thermo Fisher kits, which were collectively known as the identifiler or global filer kits, we analyze up to 16 different loci in 5 different dyes for the identifiler kits. What's shown here are the different alleles that are following the identifiler kit itself. We have 16 different loci that are identified, and as a result of that, there is a 6.18 times 10 to the minus 29th chance of having a mismatch, or having the same individual have the same allelic profile because of all the variability that's present in these different loci. The global filer kit is a little bit more advanced. It has 6 different dyes and includes 24 different loci that can be used for extra discrimination. As you can see here, you get a 7.7 times 10 to the minus 28th chance of having 2 individuals with the same global filer profile as a result of that. Now, there are some databases like ATCC and there's DSMZ in Europe that provides for cell lines ways to match some of the output of these different allelic fingerprints that you get to known allelic fingerprints for known cell lines. And you can then verify the identity of these cell lines based on the matchup of the alleles that are what you get versus what has been deposited to ATCC or DSMZ or Recon panels. For example, you can do a fingerprint of HeLa cells that you're going in culture and match that fingerprint with what ATCC says should be a HeLa fingerprint to make sure that the HeLa cells you have are in fact HeLa cells. As I mentioned, this makes it very easy then to cross-check your cell lines and make sure that the cells that are in your culture dish match up with what these organizations say have been deposited into their databases. So Thermo Fisher Scientific has, as I mentioned, a couple of different ways for analyzing these forensic samples. They're based off of the Identifiler Direct and Identifiler Plus kits. I'll just show you an example using the Identifiler Direct kit because I think this is one of the easier ways to go about doing this. The Identifiler Direct kit doesn't require nucleic acid purification. You can simply spot the cells in culture onto these nucleic cards. You take a spot then of the nucleic cards and amplify it up, analyze by capillary electrophoresis, and analyze using our gene mapper software. And to show how this works, I collected a bunch of different cell lines and resuspended them in PBS, spotted them onto the nucleic cards, as you can see on the right-hand side here. Took a little bit, a 1.2 millimeter punch out of each one of the center of the dried cards here, and then went through the identifier direct workflow. And what I was able to do is to match up using the different cell lines that have been deposited in the ATCC, match up the allelic frequencies that I would expect based on what the ATCC found. And you can see that using about 5 times 10 to the 5th cells, I get 100% match on the different instruments that we sell here. Going down to 5 times 10 to the 4th and 5 times 10 to the 3rd cells per microliter or per mil in those dilutions, I got a slight reduction in the frequency at which the correct calls are made, but I can still pick up the right alleles in those experiments. And so we recommend that you spot at least 5 times 10 to the 5th cells per mil on the copan nucleic card in your analyses. Now these copan nucleic cards are very handy because you can keep these cells in the dried state for years. It doesn't require freezer space. The nucleic acids are stable on the copan card for years, and so you can always go back to these cards then, take another punch and analyze them as necessary. This is another experiment where we mixed known amounts of M4A4 GFP cells and HeLa cells here. And this was a way to kind of see if we could detect what fraction of the cells we could detect as being contaminating in an experiment here. And in this mixtures then, what you can see by the circled red dots is that, or circle red ellipses, is that instead of having normal diploid 1 or 2 allelic representations in each one of these different loci, when there's a mixture present, you sometimes see 3 different cells as indicated by these circles here. And so then going into the different dilutions of the HeLa cells, I simply mixed them into known percentages, spotted them onto the copan nucleic cards, and was able to detect HeLa cells down to at least 10% of a mixture in that population. I could see at least half of the HeLa specific alleles that were present in that mixture, showing that the nucleic cards are very nice for even analyzing mixtures of cells. For the identifier plus cells, I purified genomic DNA from the same mixtures and was able to detect HeLa-specific loci down to about 1% of a mixture. So if 1% of the contaminating cells were HeLa cells, I was still able to detect about 1 in 5 of the HeLa-specific loci present there. And so this is a very nice way to determine whether or not you have mixture of cells or a mixture of samples present, because the specific fingerprint you generate can pick up contaminating loci down to very low concentrations. Again, the unique alleles can be detected down to around 10 to 50% of the population, in copan cards down to about 1 to 2% when you're just looking at single allelic differences in a mixed population. As I mentioned before, the global filer is another one that provides a higher level of discrimination because it has 24 different loci that can be analyzed here. And we did similar mixing experiments with the global filer cells, and we were able to detect down to about 4.6% of a mixture, detecting all the different alleles from the HeLa cell mixture there. So as I mentioned at the start of the talk, matching induced pluripotent cells with CAR T cells is very important. It's critical to be able to know that the cells that you have treated outside of the individual are matched to the donors. And so in this case here, we took some CAR T cells from some donors. They were manipulated ex-vivo, and then just to show that the ex-vivo manipulation did not change the characteristic of the CAR T cells, we matched the allelic profile using identifier kit of these different donors, and showed that the CAR T cells matched up with the T cells that we got from the donors in the first place. For the induced pluripotent stem cells, we did a similar sort of experiment where we got a series of 24 different donor and starting cells and induced pluripotent stem cells, ran a profile on them and matched up the donor to the iPSCs. In this case, when I did these experiments, they were blinded to me, and so I didn't know the identity of the samples. But by using the identifier kit, I was able to correctly match up the donor to the iPSCs 100% of the time and completely accurately. So this is described in an application note that you should be able to get in collaboration. I should say this work was done in collaboration with Dr. Wang at Stanford University. And the work describing this is in an application note that you can download from our website. And so one other important avenue of the quality control is making sure that when you're making these induced pluripotent stem cells that you either have complete pluripotency or that you have put the cells down the correct induced path here. But when you do these manipulations of cells, culturing them, you can often get aberrations. And traditionally, these chromosomal aberrations, after long passage in particular, are detected by using G banding. But this is a highly specialized and difficult process to do. One of the better ways to make sure that you haven't induced a chromosomal anomaly by passaging in culture is to do a molecular profile using the karyostat microarray. This is a way to just take snapshots across the whole genome and make sure that you're not getting chromosomal anomalies, chromosomal losses or duplications by making use of SNPs and probes across the entire genome to see if there have been induced duplications, deletions, or other sorts of anomalies. The karyostat is a low resolution way to do this, and we also have something called the karyostat HD. And in this case, using the karyostat HD and these induced pluripotent stem cells, we're able to show that the karyostat HD did pick up some smaller aberrations at multiple loci that were not present in the parental line. But because of the high resolution of the karyostat HD, we're able to find them and at least have an idea about where we had duplications or loss of chromosomal integrity in these loci. So these molecular karyotypes can reveal novel aberrations at a resolution that's better than G banding. Another thing you might want to do is to look for the oncogenic potential of these manipulated cells. By passaging them in culture, you can introduce novel mutations, and you will particularly want to check to make sure whether or not you have introduced hotspot mutation or mutations in hotspot genes that are known to be oncogenic. One of the ways this is done in a publication that we showed that we came up with was by making use of the ion torrent oncomine comprehensive assay. This is a way to use the ion torrent platform to quickly check whether or not there are hotspot mutations in a cell line. It checks for gene fusions, indels, and hotspots for 161 unique tumor driver mutations or tumor driver genes. And so in these iPSCs, we did an ion torrent hotspot analysis here, just as a subset of them looking at the P53 mutations. We didn't see that we introduced anything in the P53 locus. Similarly, we didn't see anything in the RAS or RAF or any of the other loci that are known to be hotspots for oncogenic mutations. So a quick way of analyzing the genome to see, to make sure that you have not introduced a hotspot oncogenic mutation in your manipulated cells. And then finally, the pluripotency of induced pluripotent stem cells that you've manipulated in vitro can be confirmed by using the TACMAN HPSC scorecard. This is a qPCR assay that makes use of well-known markers for either undifferentiated cells or for cells that are in an endodermic, mesodermic, or ectodermic lineage. When we look at clones of these iPSCs, we see a presence of a high presence of the of all different lineages or in undifferentiated cells. But once we move down the differentiated pathway, the EB cells show that their absence of self-renewal and lineage markers were upregulated in as a result of actually going down the differentiated treatments. This is also shown here on the graphs on the right-hand side here, where the self-renewal gene expressions are at low levels, but as a result of the differentiation, you get some ectodermal, mesodermal, endodermal pathways enriched. So the index plot shows that the self-regulation decreases and the lineage markers increase in expression once you start the differentiation process. So when you are quality controlling ex vivo cells, we recommend that there are several different things that you should be doing, and there's some best practices that we can recommend to guarantee the cells are what you think they should be. So what I didn't talk about, but one thing is that when you're working with induced pluripotent stem cells, we recommend the cells get grown in a xenogenic-free medium that supports the isolation, expansion, and reprogramming of donor fibroblast cells. This is something that our GIBCO line has provided and is easy to find. When you're looking at any of the parental and derived cell lines, we recommend using the identifiler or global filer analysis kit to make sure that the STR fingerprints match up with each other in related cells. We recommend confirming the genomic we recommend that the genomic integrity was not compromised in a manipulation by making use of the Curiostat or Curiostat HD whole genome arrays. This, again, makes sure that you haven't introduced novel deletions, duplications, loss of chromosomes of either a small portion or the whole chromosome itself. You can confirm the trilineage differentiation potential by using the TACMAN human PSC scorecard assay. And this allows you to see the expression levels of self-renewal and lineage path expression markers. And then finally, we recommend that when you are looking at CAR T cells or iPSCs, that you look for hotspot tumor-causing mutations that have been introduced by passaging the cell using the OncoMind Comprehensive Cancer Assay. It's a very simple assay that will allow you to quickly identify whether or not new mutations in oncogenic hotspots have been introduced as a result of your passage. So finally, I want to thank David Yoder for a lot of his basic work on the cell line authentication paths that we talked about. The group in the BID that worked on the iPSCs and the CAR T data, Carl Dargitz, Chad MacArthur, Mark Langdon, Uma Lakshmipathy, and Kamini Varma, under whose direction a lot of this work was done. Thank you very much for listening.